Hi there, Dr. Sadaf Qureshi here. Today we are going to talk about spermatogenesis. So what is basically spermatogenesis? Spermatogenesis is process by which sperm cells are produced in male testes, or we can say that spermatogenesis is process by which male gametes are produced by male testes. As we know, spermatogenesis process occurs in male testes, but more specifically we can say that it occurs in seminiferous tubules of the testes. If we took a cross section of seminiferous tubule and we enlarge that section, we will see that this structure look like this. This is the cross section of seminiferous tubule. These tubules are lined with very two important cells that are germ cells that are precursor to sperm cells, and the other type of cell that is fertilized cell which spore and nourish the developing sperm cell. So these are the two important type of cells that line the seminiferous tubule. The larger cells you can see there is the fertilized cell, and the smaller cells you can see is the germ cell. There's another important thing, that is the junction between the fertilized cell. There is the junction between fertilized cells that divide the luminal cavity into the basal area and adluminal area or adluminal compartment. This junction between two fertilized cells separates the basal area from the adluminal compartment, and the significance of this junction is to avoid the immune system attacking the testes. So let's talk about the phases of spermatogenesis. There are three phases in the spermatogenesis. The first phase is proliferation phase, and the second phase is meiotic phase, and the third phase is spermatogenesis phase. Now we will talk about each of these phases and we will see what happens in each of these phases. The first phase we have in spermatogenesis is proliferation phase. As we know, all of the process of spermatogenesis is going to take place in the seminiferous tubule, so we will take this diagram as for understanding and we'll see what happens in during the spermatogenesis process. The first we have is proliferation phase. In proliferation phase starts with the spermatogonia, that the precursor cells are germ cells that line the seminiferous tubule with the fertilized cells, and they are indifferentiated germ cells that are located near the basement membrane of seminiferous tubule. There you can see they both cells are spermatogonia cells, and they are located near the basement membrane and between the fertilized cells. So what happens in proliferation phase? In proliferation phase starts with mitosis that divides spermatogonia into two identical daughter cells. As there is mitosis going on, so there is no reduction of the chromosome number, so both of the daughter cells will be 2n or diploid in number. As a result of mitosis, there forms two cells. The first type of cell is type A cell, and the second type of cell is type B cell. Both of these two cells are diploid in number or 2n. Type A cells go backward towards the spermatogonia cell population and maintain their population, so the population of spermatogonia do not die away. But type B cell pass a junction area, that is the junction area between two fertilized cells, and that junction area that divides the luminal compartment into the basal compartment and adluminal compartment of the seminiferous tubule. So once the type B cell pass the junction area under the influence of chemical substance and enter the adluminal compartment of seminiferous tubule, they are now called primary spermatocytes. So at the end of proliferation phase, primary spermatocytes are formed from these spermatogonia cells, and as a result of mitotic division. Now the second phase of spermatogenesis start, that is meiotic phase. In this phase, what happens is meiosis, and that's the reason this stage is called as meiotic phase. If we enlarge this diagram, we will see that in the last phase, spermatogonia were converted into primary spermatocytes after mitosis and after passing the junction of fertilized cells. Now primary spermatocytes, as it is an adluminal compartment, it will start undergoing meiosis. And as we know, meiosis has two steps, meiosis 1 and meiosis 2. So in meiosis 1, primary spermatocytes undergo the first meiotic division, resulting in secondary spermatocytes. And this process, or this meiosis 1, ensures genetic recombination, promoting genetic diversity as there is crossing over in this phase. So at the end of meiosis 1, there are two cells formed that are secondary spermatocyte from one primary spermatocyte. Then the next phase starts, that is meiosis 2 phase. That is, secondary spermatocytes undergo the second meiotic division to form the spermatids, which are haploid cells containing 23 chromosomes. This is important that after the meiosis one phase, when primary spermatocyte convert into two secondary spermatocytes, the number of chromosomes becomes n or haploid. So after primary secondary spermatocytes are formed, there comes the meiosis 2 division, in which secondary spermatocytes convert into the spermatids, which are also haploid in number and contains 23 chromosomes in each. So at the end of meiotic phase, there is formation of four spermatids from the one primary spermatocytes. So now comes the third phase, that is spermatogenesis phase. This is the final phase, involves the transformation of round spermatid into a mature spermatozoa that are also called as sperm cells. Changes that occur during spermatogenesis phases are development of flagellum for motility and formation of acrosome, which contain enzyme to penetrate the egg during fertilization. And the third change that occur in spermatogenesis is condensation of nuclear material for streamlined structures. So in this phase, four spermatids become converted into a spermatozoa by undergoing some changes. So this was about the spermatogenesis phase, at the end of which spermatozoa cells or sperm cells are formed. There is another phase that is also called as maturation phase or epididymal maturation phase. After spermogenesis phase or after spermatogenesis, this phase come, and it is when the immature spermatozoa move to the epididymis and they undergo further maturation and acquire full motility. And this phase, that's the reason this phase is called as maturation phase, because in this spermatozoa undergo full maturation and attain its full mobility. So if we talk about the endpoint of spermatogenesis take approximately 64 to 74 days from start to finish, like to finish all the phases of the spermatogenesis. However, the continuous production of the sperms ensure that millions of sperm cells are produced on the daily basis, and this entire process of spermatogenesis is regulated by hormones such as testosterone, follicle stimulating hormone, and luteinizing hormone. In some of the next video, we will talk about how hormones affect the spermatogenesis process. So this was all about spermatogenesis. Hopefully you liked our video. Don't forget to like, share, subscribe, and comment.